Thank you for coming to my poster. Today, I'll be discussing a multimodal analysis of the transcriptional and ultrastructural states that drive the progression of pulmonary fibrosis in a mouse model. I'd like to start by discussing the clinical relevance that motivates our project, which can be found in the upper left. Pulmonary fibrosis is a global clinical burden and can result from a number of underlying pathologies. Idiopathic varieties of pulmonary fibrosis present particularly poor outcomes with only a couple years of median survival time after diagnosis. So the question that we really wanted to ask was how specific transcriptionally defined cell subtypes mediated these differential biophysical states of actively fibrotic pulmonary matrix and post-fibrotic resolution of that pulmonary matrix. And so to move on to the methods we use in our study, which can be found in the bottom left, we used a Black 6 mouse model in which these mice were intranasally instilled with either a PBS control or a bleomycin solution. And following the intranasal instillation, we characterized the ultrastructure across time, essentially modeling the progression and resolution of fibrotic ultrastructure by taking over 25 images and plugging them into a machine learning algorithm that quantifies the matrix ultrastructure. And a schematic of this process can be found in figure 2. We additionally performed microcommuted tomography as well as an assay for collagen content to support the characterizations that we found using ultrastructure. And then furthermore, we sequenced baseline fibrotic and actively resolving lungs using single-cell RNA sequencing with additional bioinformatic analysis in CellChat, SCVelo, and Cytotrace. I'd like to move on to our results, which can be found in the middle, as well as their accompanying conclusions, which can be found in the top right. So in figure 3, we began with a characterization of the ultrastructure of the interstitial matrix within mouse lungs over time. And we modeled adaptations to this ultrastructural state, or essentially the bulk changes in architecture of your pulmonary matrix using a pseudotime model. As you can see in figure 3a, with increasing pseudotime values, you have increasingly thickened fibers, more mature collagen, and disruption of the regular organization of the collagen fibers. In figure 3b, you see that at day 0, you have this baseline set of ultrastructures within a green box. After PBS administration, the set of ultrastructures observed over time remains within this box, indicating no change in the overall architecture. However, after bleomycin administration, you have progressive aberration of the pulmonary matrix. As you can see, the set of ultrastructural states gradually exit the box to a peak around day 21 to day 28, followed by gradual resolution back to baseline within that green box by about day 49. And so in figure 4a, we quantified the progression across time of that ultrastructural state and identified postoperative days 14 and 35 as active snapshots of fibrosis and post-fibrotic resolution, respectively. Figures 4b and 4c confirmed the kinetics of our pseudotime model using micro-CT and an assay for collagen content, respectively, albeit with lesser granularity than our algorithm. In figure 5, we proceeded to perform single-cell RNA characterization of the baseline actively fibrotic and actively resolving lungs, which can be seen in figure 5a. And after observing a number of pulmonary characteristic pulmonary cell populations, we used CellChat in figure 5b to identify the outgoing signaling nodes that drove differential processes during fibrosis and during resolution. And we found that macrophages serve as primary outgoing signaling nodes for fibrosis, while fibroblasts serve as primary signaling nodes for post-fibrotic repair. And so continuing these studies, we chose to isolate the macrophages and their closely related myeloid cells for a separate sub-analysis, as well as fibroblasts and their somewhat related smooth muscle cells for a sub-analysis. And so in figure 6, we performed a trajectory analysis, first in figure 6a using SCVelo, which uses RNA velocity to infer differential trajectories amongst your various subpopulations. And in figure 6a and 6b, we see that one particular trajectory of interest that we had identified is differentiation of alveolar macrophages to matrix remodeling macrophages. And this represents a highly fibrotic trajectory, given that matrix remodeling macrophages are a subtype that is most predominant during the fibrotic phase. In figure 6c, we use Cytotrace to model the differentiation order, which is on the basis of the diversity of expressed transcripts. And we observed similarly that we have a clear sort of differentiation order from less differentiated cells such as monocytes towards macrophages. And then finally, in figure 6d, we characterized the genes that were correlated with the differentiation of alveolar macrophages to matrix remodeling macrophages, given the relevance of this to the fibrotic state of pulmonary matrix. And we observed that the top 3 genes were all markers largely associated with matrix remodeling macrophages, including genes such as CTSK, which is a cathepsin that is actively involved in matrix remodeling. So all in all, this pinpoints the differentiation of alveolar macrophages to a matrix remodeling pathological phenotype as one critical differentiation trajectory during fibrosis. Moving on to figure 7, we performed a similar sub-analysis for fibroblasts and smooth muscle cells. We found in this case in figure 7a and 7b that quiescent fibroblasts served as a root phenotype for differentiation into a number of functionally defined fibroblast subtypes, including chemotactic and immunomodulatory fibroblasts, which were highly enriched during active resolution of the fibrotic matrix. These results were furthermore supported by figure 7c, which is our cytotrace ordering of differentiation. Given the importance of the chemotactic and immunomodulatory fibroblast subtypes to active resolution of the matrix, we looked at the genes correlated with the differentiation of immunomodulatory fibroblasts and found a number of genes that represent a B-cell-like phenotype, which supports that these cells essentially have an antigen-presenting and immunomodulatory function. So our overall conclusions from these studies were that the actively fibrotic state of pulmonary matrix is associated with a number of pathological matrix remodeling mesenchymal cell subtypes, and that active matrix resolution, on the other hand, is associated with a number of immune interactive cell subtypes. So in terms of some future studies that we are actively carrying out, which can be found in the bottom right, we're planning to assess the epigenomic landscape of these various cell types using single-cell attack-seq. We are currently also performing spatial transcriptomics using Visium to assess spatial relationships within the heterogeneous space of the lungs. And ultimately, our goal is to clinically correlate the markers that we may find and the mechanisms that we may find with clinical predictors of differential patient outcomes in pulmonary fibrosis. Thank you for your attention.